Hello again! Thus far into our biological journey, we have touched upon the origin of life itself, explored the inside of the single cell, and looked at how the single cell interacts with other cells and with its environment. Now, it is time to explore how we go from one cell to many cells. Here is a brief check-in question on that topic. According to the cell theory, remember that from way back when? According to the cell theory, how do new cells arise? Where do they come from? New cells arise from existing cells, specifically through the division of existing cells. So now, in order to best understand the process of making more cells, we must first understand the life cycle of any given cell. We call the life cycle of a cell the cell cycle. In all cells, the cell cycle consists of three key phases: an existing phase, a DNA replication phase, and a division phase. Each of these phases is divided into different sub-phases depending on the type of cell you are talking about, though. So let's break it down, comparing our prokaryotic cell cycle to our eukaryotic cell cycle, starting with prokaryotes. In a prokaryotic cell, their life cycle consists of three distinct but short phases. First, a growth phase in which the mass of the cell is increased, so it's growing. It is during this phase that the cell is simply existing and carrying out normal life functions while growing. Then there's the chromosomal replication phase. And finally, the division phase, where the chromosomes are separated and the cell is physically split into two independent, identical new cells. In bacteria, these are referred to as the B, C, and D periods, respectively. So, when do prokaryotic cells transition from their B period to their C and D period? Meaning, when do they transition from that growth phase to their DNA replication and division phase? This is largely related to cell size. When the cell is large enough that the surface area to volume ratio is no longer efficient for the cell replicates its DNA in preparation for cell division. The time a prokaryotic cell spends in its B period can vary anywhere from under half an hour to over several days. Although most bacterial cultures in laboratory settings or in a nutrient-rich environment have generation times of under just 1 day. Like prokaryotes, eukaryotic cells have their own existing phase, a DNA replication phase, and a division phase, but these phases are divided a bit differently. Let's look at what I like to call this existing phase. In eukaryotes, we call this interphase. Now, within interphase, eukaryotes have their DNA replication phase. This is distinct from what we saw in prokaryotes, right? And interphase is divided actually into three subphases. We have our gap1 phase, abbreviated G1. We have our synthesis phase, abbreviated S phase, and our gap2 phase, which is abbreviated G2. Now, during G1, the cell is simply growing and existing, carrying out its primary functions. For instance, a lung cell is working to help the organism with breathing, or a stomach cell is producing acid to aid in digestion. Some organelles are also replicated, or they replicate themselves during this phase. Remember, certain cell types require a higher number of certain organelles compared to others, right? For example, cardiac muscle cells, those require a lot more ATP and energy than other cells. So these cells replicate a lot more mitochondria within them in order to meet that high demand for producing ATP. During S phase, DNA synthesis occurs, meaning the DNA within the cell's nucleus is replicated. This is one of the several places where eukaryotic and prokaryotic cells truly differ. You see, in prokaryotes, Once the DNA is replicated, the prokaryotic cell automatically begins its cell division process. In eukaryotes, however, the cell does not go straight into cell division after DNA replication. There is another gap phase that takes place after DNA replication occurs. This second gap phase is super important though, because remember, unlike prokaryotes, eukaryotes have organelles. If the cell is about to divide into two new cells, we definitely want to make sure that the new cell has the organelles that it needs, right? So in the G2 phase, some more organelles are duplicated, and the cytoskeleton is dismantled to provide resources for the division phase. The cell also replenishes its energy stores to synthesize proteins necessary for the chromosome manipulation and movement that will occur when the cell divides. And there may also be some additional cell growth during G2. But basically, G2 is used to ensure that the cell is fully prepared to enter its cell division phase. So, once the eukaryotic cell is ready, it enters its division phase. In eukaryotic cells, we call that division phase the mitotic phase, or M phase, whereas in prokaryotes, we call the division phase binary fission. The mitotic phase is broken down into two overarching subphases, mitosis and cytokinesis. Mitosis is the separation of replicated chromosomes and the division of the nucleus, while cytokinesis is the physical process of cell division, which divides the cytoplasm of an original cell into two new cells. Mitosis is also subdivided into 5 subphases, but we'll touch on those later. For now, let's zoom out and compare these two cell cycles as a whole. First reminder I want to make is that all prokaryotes are all single-celled organisms. We do not have multicellular prokaryotes. They are all just single-celled organisms. One cell is its own living entity. Similarly, eukaryotes can also be single-celled organisms, where one eukaryotic cell is its own living entity. However, eukaryotic cells can also contribute to multicellular life. All multicellular organisms are made up of eukaryotic cells. Now, in single-celled organisms, a single-cell division cycle is how the organism replicates itself. It's basically how it reproduces. It reproduces by making a clone of itself through cellular division. In multicellular organisms, though, such as plants and animals, a series of cell division cycles is how the organism develops from a single-celled fertilized egg into a mature organism. It is also the process by which hair, skin, blood cells, and internal organ cells are regenerated and healed. After cell division, each of the daughter cells begin the interphase of a new cell cycle. Or in a prokaryotic cell, they re-enter that B period of their cell cycle. Now, just to clear up some verbiage that I've been using before moving forward, you've probably been hearing the terms parent cell and daughter cell. Let's break that down. When single-celled organisms divide to make two new cells, the cells produced are exact copies or clones of the original cell, right? Biological parents provide genetic material to their offspring. So, in essence, the original cell is basically the parent to the two new cells formed through cellular division. Therefore, when talking about cell lineages, even within multicellular organisms, we refer to the original cell as the parent cell, and we refer to the identical cells produced through cellular division as the daughter cells. Make sense? Before wrapping up this brief overview, I just want to touch on one last thing because it's actually pretty darn cool. This may seem somewhat surprising, but certain organelles in eukaryotic cells actually go through the process of binary fission, just like prokaryotic cells. Pretty cool, right? So, checking question. Based on what you know about cell structure and the theory of endosymbiosis, what organelles do you think undergo binary fission within a eukaryotic cell? Well, as it turns out, just like prokaryotic organisms, chloroplasts and mitochondria both contain DNA packaged as single circular chromosomes. These organelles also replicate their chromosomes and undergo binary fission within the cytoplasm of eukaryotic cells. Information such as this, along with some other similarities shared between these organelles and prokaryotic organisms, support the theory of endosymbiosis. Pretty cool, right? So this is the overview of the cell cycle and how a single cell can divide to make new cells. Enjoy the rest of the unit.